Explain the mechanisms within meiosis that generate genetic variation among the products of meiosis. So there are two main mechanisms, the first one being crossing over. This happens when two attached homologous chromosomes, in other words a homologous pair, swap genetic information. This means that a maternal chromosome can end up with a segment of genetic material from the paternal chromosome, and vice versa. Crossing over occurs in linked genes. The second mechanism is called independent assortment. Genes found on different chromosomes show independent assortment. Independent assortment of genes on the different chromosomes occur because during metaphase 1 of meiosis, tetrads, also known as pairs of homologous chromosomes, line up along the equatorial plate with a random orientation. The random orientation of bivalence during metaphase 1 causes variation in the way different alleles on different chromosomes are grouped together. The independent assortment of each chromosome during meiosis gives it an equal chance to end up in either daughter cell. Explain the implication of crossing over on linked genes. Linked genes are genes that are located on the same chromosome. Through crossing over, the alleles of these linked genes get swapped between the homologous chromosomes that carry them. This can lead to the formation of chromosomes in the daughter cells that will not have the same combination of alleles of the chromosomes of the parent cell. These shuffled chromosomes are also called recombinants. The implication of crossing over on linked genes is a higher genetic variety in an observed population. Number 3. What impact will crossing over have on patterns of inheritance related to linked genes? So basically, when linked genes cross over, they transfer alleles. They will result in the same chromosomes having shuffled genes because they do not have the same combination of genes as before. The process ends with recombinant gametes, which leads to more variants in the DNA that the offspring will inherit.